Thank you, Lisa. For those who didn't see my talk yesterday, I am Adeline. I'm based out at the Childhood Dementia Research Group at Flinders University, and I've been researching Sanfilippo for many, many years now. It's my pleasure to introduce our first speaker today. I believe he's joining us online, and I think I saw his name come up in the chat, so I believe he's here. Professor Mark Walterfang is a consultant neuropsychiatrist at the Royal Melbourne Hospital and an expert in managing comorbid psychiatric and neurological disorders, neurodegenerative diseases, neurometabolic disorders, and atypical dementias. He was involved in the establishment of the Melbourne Young Onset Dementia Service and manages a large cohort of adult patients with the rare neurometabolic disorder Niemann Pick Type C. Dr. Walterfang has also been involved in clinical trials and the development of clinical tools for the use of psychiatric patients in the areas of cognition and behavioural observation. If you're there, Mark, if you're able to turn on your camera and you can start sharing your slides when you're ready, you can take it, start whenever you're ready.
Thanks so much for inviting me, and thank you very much for having me speaking. I'm just going to present our slides today. So I'm a neuropsychiatrist with an interest in neurodegenerative disorders, particularly young onset neurodegenerative disorders, which does generally include a number of neurometabolic disorders. My main interest, as you've heard, has been in the neurometabolic disorder Niemann Pick Type C disease, where we manage a large adult cohort of this group of patients and have done for about the last 25 years. What's that has meant, I imagine most people understand, if you've got a big enough cohort, you can start to study this group of patients quite extensively, but also it's great to have the capacity to have enough for clinical trials. So we've had about 25 patients over that period in time, so we've been able to do a range of different studies, which I won't be talking about today. We've also been using disease-modifying therapies since about 2008, since we first had access to miglustat, also looking at cyclodextrin. We've been running both intrathecal and intravenous trials of cyclodextrin. And more recently, been looking at Acetyl Leucine, which is a very interesting compound, which I'll talk a little bit about today, and something that may have some relevance for Sanfilippo syndrome as well. So just disclosures. Most of my disclosures are about involvement with pharmaceutical companies, particularly those involved in the manufacture of antipsychotics or treatments for Niemann Pick Type C disease. So we'll talk a little bit about N-acetyl L-leucine. So it's a modified version of the amino acid leucine. Now, this has been around for quite a long time. The racemic mixture of the D and L, the left and right-handed molecules, has been around since 1957 in France as Tanguanil. It was a treatment for acute vertigo, which I understand was not a very good treatment. Although this slide has said it hasn't been approved in any other jurisdiction, it has had periods of approval in Vietnam and a couple of other countries. But really now, Tanguanil remains available in France. And this is something that many NPC patients have been on when they were looking for access to anything that might change their illness presentation, reduce symptoms, or even slow the progress of their disease. And indeed, many of my patients have been on Tanguanil for 5 to 10 years. However, the L enantiomer is clearly, and this has been demonstrated in a number of studies, it seems to be the more active enantiomer and is much more, not just biologically active, but appears to be neuroprotective. And we'll talk a little bit about this. There's been some pretty good evidence that it doesn't just improve symptoms in NPC, which it does fairly rapidly, but it may have a disease-modifying effect demonstrated perhaps most convincingly in the mouse and in the open label extension of the current trial, hopefully in human patients as well. So to talk a little bit about it, so L-leucine is an amino acid that has powerful signaling properties. Now, this is usually taken up by the L-amino acid transporter, or LAT. However, that's a relatively slow system that has limited uptake, and it's kind of readily saturated. However, acetylation of this allows it to use a couple of different transporters, in particular MCT, also a couple of OAT transporters, and that has a much higher capacity, and that allows a lot more acetyl-L-leucine to be delivered effectively to all cells and to have its metabolic effects, particularly in the brain, also the liver and spleen, which are organs affected by most lysosomal storage disorders. And what's interesting is that it appears to improve mitochondrial function and lysosomal function perhaps secondary to improvement of mitochondrial energy utilization. It does restore membrane potential in terms of its interaction with phospholipids, and appears to dampen neuroinflammation. So it's an interesting little phalanx of mechanisms of action that it has. And overall, it looks like, and the hope certainly is, that it can prevent cellular death. There's some fairly good evidence in animal traumatic brain injury models, and by dampening neuroinflammation, improving mitochondrial energy utilization, that it might show improvement in a range of lysosomal storage disorders. So this drug, I mean, certainly my patients who are on our clinical trial, and we have 4 patients who are in the open label extension phase, it's readily administered orally. It's 5 grams, which is study doses, in a couple of sachets that get mixed into orange juice or water. So it's easily administered orally a few times a day. It's got a relatively benign safety profile. And I think the interesting hope for us is that NPC, which is the progenitor disease where it's been showing benefit in studies for about the last 10 years, since Tatiana Bremova first published, I think it was a case series in neurology in 2015, showing how it benefits. That has kind of slowly developed into a more formal series of phase 2 and 3 clinical trials, the last of which I'll talk about. So it improves mitochondrial function, it secondarily reduces oxidative stress. It does appear to normalize membrane potential and cellular signaling, and perhaps secondarily improves lysosomal function and reduces substrate accumulation, lipids, potentially gangliosides as well. Okay, as I've mentioned, it's a little bit reduplicative. So let's talk a little bit about what we've seen in some of the studies. So in untreated NPC mice, 59 days of age, significantly decreased levels of pyruvate dehydrogenase E1 alpha and beta and binding protein. But it certainly seems that when you add N-acetyl leucine, we get a significant restoration of this, which we can see in the left-hand sided figure. Also, potentially elevated oxidative stress because of radical oxygen species contributes to neurodegeneration across a range of different disorders, not just the lysosomal storage disorders, but the late onset neurodegenerative disorders, particularly Alzheimer's disease and Parkinson's disease. And indeed, some really interesting early clinical data suggests that NALL does look like it has some benefits in early stage Parkinson's disease, and it seems to mediate reduction in these ROS levels and normalizes glycolytic pathways. And that's been shown in Sandhoff and NPC mice. Now, the increase in lysosomal functioning looks like it's secondary to an improvement in energy metabolism. We know that an expanded lysosome appears to be a pretty reasonable marker of lysosomal dysfunction. And so that's been the proxy measure that's been looked at in a number of these studies. And we can see that when we get the treatment with NALL in the mouse certainly reduces the expanded lysosome. You can see here in the left-hand slide, that expanded lysosome gets significantly reduced with this, suggesting that there's the potential to look at beneficial effects across a number of lysosomal storage disorders. We also know that neuroinflammation is a hallmark in NPC. This has been shown convincingly in the mouse model. We demonstrated this in vivo studies using PET in our human population of adult NPC patients, showing that neuroinflammation looks like it's not just an epiphenomenon or a bystander in NPC. It looks like it may well significantly drive pathology. And in this mouse study here, the significant elevation of neuroinflammation that was seen in the mouse brain, particularly in the cerebellum, which is in NPC, one of the hotspots for neuronal loss, the microglial activation significantly reduced. And it does look like these mechanisms of action correlate to symptomatic and neuroprotective effects. Certainly when the drug was first studied, the thought was maybe it just increases cerebellar glucose metabolism. That's why we seem to see such a relatively rapid improvement in our patient groups. But it looks like it's probably doing more than that. The motor dysfunction that we see in the NPC mouse that becomes apparent at 6 to 7 weeks of age gets rapidly corrected symptomatically with now, but pre-symptomatic treatment appears to delay the onset of this motor dysfunction. And this is the putative neuroprotective effect that early treatment may well slow the course of the disease. And in all neurodegenerative diseases, no matter whether you're looking at early onset, childhood onset, young adult or older adult onset, this is absolutely the holy grail of being able to find something or even a suite of different compounds that can do that. Certainly in NPC, we see a slowing of disease progression in patients on miglustat, and it looks like we're getting a similar benefit from NAL as well, or IV1001 as the study compound is called. And certainly in the mouse model, it's been demonstrated that miglustat and NAL appear to have a synergistic effect. But even the mouse treated with NAL alone appears to have a significantly increased lifespan. And again, this looks to be, it's related to the L enantiomer rather than the D enantiomer. So the L enantiomer has an effect, D does not. A mixture still has an effect, but the effect is somewhat blunted with the racemic mixture or the tangenol, which has been around for so long, largely because the D enantiomer appears to also competitively, it competes for uptake across the transporter. So we've seen that it does significantly reduce neuroinflammation. We can see here this on the left-hand side, this is from the NPC knockout mouse. It's also been demonstrated quite conclusively in a mouse model of traumatic brain injury as well, that there's a significant reduction in neuroinflammation and neuronal loss. A much older study on the right, which was looking at a guinea pig, showed that the racemic mixture or tangenol really stabilized cellular signaling. Apologies here. So with the NPC mouse model, here we can, the untreated mice at 9 weeks of age in vivo, when they're treated, there's significant and noticeable improvement, not just in their neurological functioning, but also in their body weight. You can see one pretty happy skippy mouse and one much less happier skippier mouse. And this has been shown not just in the NPC mouse model, but also in Sandhoff as well. So we see significant improvements in and basically two specific gangliosidosis that have a significant accumulation. The thing that NPC and Sandhoff have in common is the accumulation of GM2 gangliosidosis. This was the initial study driven, Fran Platt in particular from Oxford, but also Michael Strupp, the neurologist from Munich, really drove some of this work into the difference between the two enantiomers and how different L is. Some of the early mouse studies, this was published in 2020 by H.M. Kaya, showing a significant reduction in disease progression that appeared to be relatively specific to the L enantiomer rather than the D enantiomer. A combination worked, but the L enantiomer seemed to be where the effect was captured, as we can see in the bottom row of slides here. So this is one of the GM2 gangliosidosis patients. And so this was the first, this was one of the publications of this. This was looking at the L enantiomer and Niemann Pick type C disease, basically showing in the phase 2 trial, looking at the scar functional index, the SCFI, the significant improvements that we saw in the treatment period versus the washout period. So the interesting thing about this is the rapidity of improvement happens over a relatively short space of time. And the trial design that ultimately was successful was a crossover trial design with only a couple of months treatment, and then vice versa, and then moving into the open label phase. So this was the pivotal trial. 60 subjects were enrolled and randomized, and in this trial, the SARA was used for ataxia, as well as the SCFI, clinical global improvement, the modified DRS, and quality of life scales. So, and really, what did we see? There was, it's an interesting design for rare disease studies in terms of not necessarily needing a placebo group. We're also running another trial of another compound in NPC that's quite a long trial that has a 2-year period of placebo control, which is actually being something that's not straightforward for a patient group to sit on something and not know whether it's they're on active compound or not for 2 years before they move into the open label phase. But really, the compound showed a significant and meaningful improvement versus placebo on primary and secondary endpoints, improving just a range of symptoms, particularly measures of ataxia, but also cognition, behavior, and mood. And I'll talk a little bit about some of our observations of that, and a significant improvement in functioning. And it appeared to be irrespective of what age group, adults or pediatric patients, and whether or not you're already on miglustat, because worldwide, it's really quite hard to find patients who are not on miglustat. So here you can see the 3-month crossover. So you're either active or placebo 3 months after your washout period, and then swap another 3 months, and then into the extension phase. So again, just to reiterate those key findings, significant improvement on the primary endpoint, which was the SARA or modified SARA, but also secondary endpoints, appeared to improve many symptoms. And when it was replaced with placebo, when you started active and then went to placebo, there seemed to be really, there was a significant and noticeable worsening of neurological symptoms. For our clinical trials group, it was interesting as we were running this trial concurrently with another compound, and everyone had thought that, well, we don't really know. The other compound was intravenous. We'd previously run intrathecal trials. And the general sense was, well, what is an oral powder going to do? It just shows you how our inherent biases can affect how we think things are going to change. Surely the intravenous compound is going to work better. But we were really struck by the rapid and clear improvements in patients, even though, of course, our fellows were blinded throughout. They were saying some people have improved really markedly, much more than we expected. The drug was really well tolerated, had no serious adverse reactions. And really, this pivotal trial, the results were fairly consistent with the earlier phase 2b trial. So it met its primary and secondary endpoints really quite nicely, but particularly the primary endpoint. And we could see that this is really looked like it was significant across all subgroups. And you can see that the pediatric and adult patients all showed significant improvements. Okay, so here are some quality of life with some feedback. And what was interesting is what seemed to come through was not just motor improvements, but also I think they're probably attentional improvements, awareness improvements, which can be a double-edged sword, which we'll talk a little bit about. But people just felt that our NPC patients were just much more present, could be more independent. Now that can be a real shift for some family members that when even some of our family members describe people as seeming almost a little bit more feisty. They wanted to push more towards independence, and they were really, really struck by this. The safety profile was relatively compelling, nothing that occurred in more than 10% of patients, and only a couple as seen as related to the drug compound, constipation, restless legs, which is interesting because there's some interesting early clinical data that the drug may actually be quite useful in restless leg syndrome. So it does look really quite benign. And as people are aware, this was likely aware, this was published a couple of months ago in the New England Journal of Medicine. Just reiterating that data there. You can see the adverse events not really significantly different. So lastly, our impressions as a trial site, as I've mentioned, we were really quite struck by some of the early improvements, the rapidity of improvement, not something that we'd seen previously with miglustat in this patient group, but after a few weeks of treatment, improved alertness, engagement, and for some people that meant waking up from apathy and hypersomnolence to being just wanting a bit of get up and go, but potentially also that being refracted through pre-existing cognitive issues like inattention, impulsivity, etc. The motor scores invariably improved. So when I was approached to talk a little bit about our experience with this compound, which surprised and delighted us as well as patients and their families, to talk about what the relevance might be to Sanfilippo disease. You know, it does share some features with NPC. It's a lysosomal storage disorder. There's lysosomal dysfunction, accumulation of gangliosides, as well as GAGs. There's expanded lysosomal volume. There's a significant neuroinflammatory component. There's impaired autophagy, lysosomal functioning, and progressive cognitive and motor impairment does seem to correlate with cortical and cerebellar pathology, at least as measured by volume loss on the imaging studies that I'm aware of, and imaging is really my research focus in neurological disorders. So there has been a little bit of data from MPS IIIB patients. These were patient fibroblasts that have been looked at. And what you can see here, and there's been, this is unpublished data, but there's been some evidence that this patient fibroblast line, that there was a significant increase in fluorescence consistent with an expanded lysosome, but with treatment that this improves significantly. One of the really interesting things for us is, you know, when you translate cell line data and start to think about translating that into really an available treatment is, you know, drug delivery. But simply this drug, one of the interesting things is through the acetylation shifting its transporter that makes it so readily absorbed and taken up so quickly in comparison to the non-acetylated version. It makes it really, really quite attractive to think about in these disorders. The flip side to all of this as I close, so this is a really interesting compound. It's very tolerable, oral, rapidly absorbed, gets right into all the tissues that we want it to get into. It looks like it has a broad mechanism of action. Clearly, our kind of index group of patients with MPC showed significant improvement. At the same time, this is also a very small company with limited resources that has been looking to see if it can get an indication. And if the company is able to get an indication for NPC off the back of this trial, then it'll be very interesting to see where to next. The company does have early preclinical data, as we've seen, in different cell lines and different mouse models. And certainly in my discussions with them, when I did mention, look, I've been asked to talk a little bit about our experience and the relevance that there may be for Sanfilippo patients. I did say, well, is this something you guys would be interested in? And they certainly said, look, we're interested in just about everything. It's about resources and priorities, but they're certainly interested in feedback from the Sanfilippo community on interest in this compound, potentially whether anyone has ever used Tangenol, because that's, you know, of great interest to them. They're focused on the lysosomal storage disorder community. And I'm hopeful that the NPC patient group demonstrating such clear benefit will be something that we can see in other groups over time.
Thank you, Mark. That was some beautiful data presented there. Are there any questions from the room, or if there are online, if you want to put them into the chat box?
Hi, Mark. Lisa here. Thank you for a great presentation. It's really interesting and exciting to see that data. I was interested in the idea of the combination with miglustat and the acetyl leucine. Do you foresee needing to do a clinical trial specifically to look at the synergism of the two compounds, or were you able to see that? I wasn't able to see in the subgroup analysis that you did. Did you look at whether there were was anything in that subgroup analysis of the people that were on miglustat and not on miglustat?
Look, can people hear me? Yeah, the general sense is that there wasn't a great deal of difference. However, the hope is with the extension phase, we've now had, everyone's been on active compound now for 12 months. The hope is that with more data over time, we'll be able to tease that out a bit more. It certainly did. You know, the miglustat's an interesting compound. It's a substrate reduction therapy, and you know, it certainly does slow the disease, but it has really, by and large, one mechanism of action, which is reducing GM2 and GM3 gangliosides. And there's so many other things that happen in NPC cells that it doesn't necessarily address. And there's certainly been data published for many years in cell lines, but also the mouse model showing it having synergistic effects with different compounds. Even ibuprofen was one of them. Adrobetidex or cyclodextrin has been another. What's been really tricky, it hasn't been looked at with ibuprofen, has been looked at with adrobetidex, was actually trying to translate that into you know, evidence of a synergistic effect in humans. So that's been difficult. I don't think really we actually have enough data yet, at least on the basis of that trial. Hopefully the extension data will show us that. I know that this company is very interested in looking at that 12-month mark and seeing if more can be teased out. The tricky bit, of course, is, you know, and I know talking to Mark Patterson, who's probably the guru of treatment in NPC and in a range of different metabolic disorders, he says that his sense is, you know, until we're really able to correct the genetic deficit, you know, for many of these disorders, it's going to be combination treatments. Really, really tricky to convince regulatory bodies that that's actually worth funding, particularly when individually these drugs can all be very high-cost drugs. So, yeah, the answer is no, not yet. Hopefully soon, but I'm not really sure that that's necessarily going to help us, if that makes sense.
Okay, I think we'll take an online question and then we'll take the last question in the room.
Online question has just been answered.
Excellent. All right.
Sure.
Thanks, Mark. That was fantastic. There's definitely an interest in this in the Sanfilippo field, so it would be great to have discussions.
And I'm happy for anyone to contact me or for me to connect people with the company at any stage as well.
That would be great. I'm wondering, the mouse studies you showed were pre-symptomatic treatment, yet the patient video is clearly some sort of reversibility of already existing symptoms. I'm wondering if you can speculate on, you know, whether that's likely to extend, you know, for the life of a patient. Is there a point of no return, do you think?
That's a really good question. That is a really good question that I've thought a lot about. I mean, we started treating patients with miglustat in 2015, our adult patients with NPC. And what's striking to me about this experience is what we've done is we've initiated a clearly disease-modifying treatment at very different points in people's symptomatic journey. I actually have a patient who is a working dentist who has now been on miglustat for not quite 15 years, and she is still a working dentist. She has had some decline, but it's been incredibly subtle. And just by sheer fortune, we were able to start this person on drug at an almost pre-symptomatic stage. Whereas I've had other people who've been symptomatic for 10 years before we were, and it was just, you know, when the drug became available to us on compassionate access. And so what we've been able to see is that when you introduce the drug in terms of their disease progression, does play a significant role in the reversibility. And it makes logical sense, I think, to think about that there are obviously some irreversible cellular cascades that can maybe only be partially arrested, or that maybe at a certain point, particularly in neurons, that you cannot rescue, even if you dampen down neuroinflammation, etc. So I think that probably applies to all disease-modifying compounds in any neurological or neurodegenerative disease. The interesting thing about N-acetyl Leucine is that I think it has very early metabolic effects that are probably at least semi-distinct from any neuroprotective effects that it has, which are probably a bit broader spectrum. And so, early effects maybe about kind of mitochondrial functioning, maybe about, translating to glucose utilization, neuronal glucose utilization, maybe about stabilizing membranes and hyperpolarization, and then much later effects that may be about, there's some evidence about autophagy, we've seen the evidence about neuroinflammation. So that early bounce and then hopefully a slowing of progression is what I think we can reasonably expect to see. Having said that, when we started treating patients with miglustat, which was in 2008, we actually saw for some of our patients who were quite symptomatic, like the woman with GM2 gangliosidosis who was on that video, that we saw improvements over about 6 months, and then again, a resumption of disease progression, but what looked at clearly a significantly slower rate. As I've mentioned, our studies were mostly in neuroimaging, and so I was really interested in capturing neuroimaging indices of disease progression. And we certainly showed that for people who were on miglustat versus those who were not, there were like cerebellar volume, white matter integrity was different between those groups of patients. So I think, when we've got really good evidence about things being disease modifying, the earlier in the process you can start the drug, the better. I know that's kind of a discursive answer to your question, but I think I'm just trying to capture that experience we had with miglustat, but also how IB1001 or N-acetyl Leucine may be different in, I could talk, there's another reference disease I think about, which is PKU and effects of dietary modification on that. There are changes to kind of neurodevelopment, but also much more immediate state-related changes. And that's what I think we see with IB1001.
Thank you very much. If everyone can join me in thanking him again for our presentation. So our next speaker is also an online presentation. We have Dr. Johannes Strzelecki joining us from San Diego. He's a clinician scientist who trained in Germany and now works at the University of California. As a neurologist, he specializes in neurodegenerative diseases and the diagnosis and treatment of movement disorders. His research includes neuroimmunology and epigenomics in the context of neurodegenerative diseases and the development of disease-modifying treatments.
Thank you so much for the kind introduction, and it's a real pleasure to give a talk about my favorite cell type, microglia. And over the last 1.5 days, we already heard a lot about neuroinflammation, and probably the big part or major part actually that also neuroinflammation plays in Sanfilippo syndrome. So it's not only neurodegeneration, but maybe also microglia and astrocytes that could drive the disease pathogenesis. And my lab is really interested in understanding how microglia work, what are they doing in the context of Sanfilippo syndrome, and how can we tweak them to be beneficial in the course of the disease. So most of the work has been done by a fabulous and talented PhD candidate in my lab, Chris Ballack. He's graduating next year. And he did wonderful work on Sanfilippo syndrome. And so most of the stuff that I show is here. This work could have not been done without with the help of the Sanfilippo Children's Foundation and CERN, which fund the project that I talk about today. So let me start a little bit talking about microglia. These are the innate immune cells of the brain. They had like a boring name, resting microglia, as if they do nothing in the brain. But here I show a picture of microglia, how active they are actually in the brain during homeostasis. So these are human microglia, and you can see the cell body in the middle, and they have a lot of branches. And they always constantly survey their microenvironment. They have contact to neurons, astrocytes, oligodendrocytes, and so on. And they are very plastic cells. So they respond to injury. So once they detect a pathogen or protein aggregates or anything else, they respond dramatically. And this is also shown here. So in the middle, again, human microglia. In red, you have neurons, not so important. Here, they're just bystanders. And then I would like to draw your attention to the middle of the screen. Here, there will be a laser injury. So we injured a vessel to see how microglia respond to injury in the brain once they detect injury. So they're still constantly surveying their microenvironment. And once there's an injury, they then branch out to the site of the injury and then try to then be restorative or beneficial, remove the debris, and so on. This was work done with the lab of Rusty Gage, who's also very involved in the Sanfilippo syndrome projects that I showed today. So what are the core functions of microglia? They're innate immune cells of the brain. They have a lot of functions. Once they detect any changes during development or during disease, so they can phagocytose, take up, remove synapses, or they can remove dying neurons. They can respond to pathogens. Very important for Sanfilippo syndrome, they can respond to protein aggregates or accumulation of storage disorders. And what they do is they release soluble factors like TNF, interleukin-1 beta, or also anti-inflammatory cytokines like interleukin-4 or interleukin-10, depending on the context, in response. And so they have a lot of functions. So they're not only phagocytose and secrete factors, but they're then really involved in neurogenesis, so generation and removal of new neurons. They're involved in removal of synapses. They're very important for neuronal function. So I was very intrigued yesterday to learn about Cedric's work now really integrating astrocytes and neurons in the brain dish. They're very important for tissue repair. They play a role in inflammation, myelination, and so on. So how does it then work in the context of neurodegeneration or in the context of Sanfilippo syndrome? So we don't really know. Are microglia very beneficial or are they neurotoxic? My opinion is that early during the course of the disease, they're beneficial because they remove protein aggregates or heparan sulfate, like glycans, secondary storage products. So they clear, they remodel the tissue, they're neurotrophic, so they keep the neurons alive. They can be restorative. But during the course of the disease with chronic inflammation and accumulation of heparan sulfate and glycans and so on, this can then actually lead to demyelination, so they impair the work of oligodendrocytes. They can then also activate astrocytes like shown yesterday, so astrocytes can also be inflammatory, and eventually this leads to neurodegeneration. Also, what we know in Sanfilippo syndrome, there could be an accumulation of amyloids, what we also see in age-related diseases, in Lewy bodies, so aggregates of alpha-synuclein, and so on. So it's very interesting to study microglia, what are they actually doing in Sanfilippo syndrome? And one way is really then to find out what are the molecular mechanisms, or what are the molecular phenotypes of microglia and MPS IIA. So I caught really my attention here through the work of Jeff Esco and Philip Gortz here at UCSD, who were studying MPS IIA. They developed a mouse model for MPS IIA. It's a little bit different than most other models. So they have knocked out exons 3 through 6 of the SGSH gene that encodes alpha-sulfamidase. And what this then leads to is that this enzyme doesn't have any activity. So there's no enzymatic activity of sulfamidase in this mouse model. Also, they have accumulation of heparan sulfate and glycosaminoglycans from birth on. The lifespan is reduced. So usually the mice live roughly 1 year, and they also have a behavioral phenotype. So they become first more hyperactive, but later also have motor symptoms and also cognitive problems. And so my lab is doing a lot of genomics, so I'm interested in transcriptomics, so looking at gene expression, also at epigenetics, what I don't show today. So epigenetics, looking at DNA methylation, and also in histone acetylation, transcription factors that drive then phenotypes of microglia and other cell types. And we also do immunohistochemistry and electron microscopy. So here we were isolating microglia from the brain of those mice. So first we started out with immunohistochemistry. So how do microglia look like in a normal brain and also then in the MPSIIA mouse model. So in red you have EBA1, that's a classical marker for macrophages. And you can see here that the microglia have the typical morphology. So they have the small cell soma and then a lot of branches with they survey their microenvironment. Usually under homeostatic conditions, they don't express CD68. So CD68 is a lysosomal marker, and when a microglia becomes activated, they upregulate CD68. In this case, you don't find CD68, and this is a bigger magnification. What happens then in the lysosomal storage disorder in the mouse model for MPSIIA? You see then here in the bigger picture, the EBA1 positive microglia cell. So the soma became bigger, the branches are a bit smaller. So that's already indicative of microglia activation. And you can see then also in yellow, CD68. So those microglia became activated. They have a lot of lysosomal activation. So we call them reactive microglia. We then went a step further and looked at electron microscopy. And I think in the first or second talk yesterday, there was also a beautiful electron microscopy picture from 2005 that really showed big enhancement of lysosomal swelling. So again, here in the top row, you have the wild type mice. So here in dark, this is the nucleus of a microglia. So microglia have a more darker nucleus, a little bit bigger, and more bright are the nucleus of neurons. And what you can see here already in the MPSIIA mouse model, you see this big blobs, and these are lysosomes. So we have big lysosomes, especially in the microglia. You can also see some in the neurons, but not as pronounced as in microglia, really showing that microglia show a lot of this lysosomal swelling. And this is a bigger picture of the microglia. It's really bigger, and you see a lot of those lysosomes now. We then sorted microglia, so these here are sorted microglia, and you can see also the big difference between the wild type and the diseased microglia. So we have a lot of lysosomes here in those microglia. We then were isolating nuclei from the brain of MPSIIA mice. So this is a single cell RNA-seq, UMAP, what we call it. So every dot is a cell. So here, for example, like lila, you have neurons. These are GABAergic neurons. We can identify those by the upregulation of certain genes. You have a lot of oligodendrocytes. So in blue here are oligodendrocytes based on marker like myelin basic protein and so on. You have astrocytes. You have oligodendrocyte precursor cells. You have excitatory neurons, so glutamatergic neurons. And you can also see a very small population of microglia. So roughly 5% of the brain cell types are microglia. We were then interested in how does the gene expression program change in the different cell types. So to our surprise, we saw a lot of changes in microglia, but not so much in the other cell types. So you saw some changes in the glutamatergic excitatory neurons and also in the inhibitory neurons, but not as pronounced in the microglia. But that's one very characteristic feature of microglia. They really respond to injury in the brain. Whereas neurons, they change their function. So you will see, like yesterday, there's differences in synaptic function, there's neurodegeneration, but usually we don't detect so much changes in gene expression. We were then isolating also microglia to get more microglia because they're only 5% of all brain cell types. To detect more microglia, and you can see here in blue the wild type microglia, so from a normal control mouse, and then in orange are all the ones from the MPSIIA mouse, and you can see a clear distinction between wild type and MPSIIA microglia. You can then look at different clusters of microglia. So each color here in the middle are different clusters. So they are homeostatic microglia, for example, here in blue. And this is predominant, the microglia that you find in wild-type microglia. But you find also a cluster of microglia that you don't find in wild-type and that are only detectable in the MPSIIA mouse model. And this is here shown in red. So we have now a predominant microglia cluster that is associated with the microglia or with the MPSIIA phenotype. We were also looking at a time course, so when can we actually already detect changes in gene expression in microglia? And as I told you, that already at birth we can detect glycosaminoglycans, heparan sulfate accumulation in those mice. And already at 3 weeks, we detect massive changes in gene expression in microglia, and this is progressive. So at 4 months, you have already 900 or so genes that are differentially expressed between wild type and MPSIIA mice. And this allows us really to look at what are the gene expression changes in microglia during the course of the disease. So the bar graphs are showing changes in gene expression. So you have here in red wild type numbers, and here in blue the gene expression of MPSIIA microglia. You can see nicely here between 3 weeks and 8 months, you see a stark increase in interleukin-1 beta, and that's one of the pro-inflammatory cytokines that we know can actually lead to neuronal dysfunction, then also can lead to neuronal death. And there are also clinical studies going on targeting actually interleukin-1 beta. And so we could also detect this in the mouse model. Another cytokine very prominent is TNF-alpha, also pro-inflammatory cytokine that can induce neuronal dysfunction and neuronal cell death, which was also progressively upregulated in MPSIIA mice. The homeostatic genes like CXD-CR1, TMIN119, so these are classical microglia housekeeping genes, I call them. These are downregulated. So this really shows that microglia become activated in MPSIIA. We see other genes that are associated with a neurodegenerative phenotype. The prominent example is ApoE, very famous for Alzheimer's disease. Also known to be upregulated in Alzheimer's disease, but we also see a dramatic increase in gene expression in the MPSIIA mouse model. TREM2, another target in Alzheimer's disease, is also upregulated in MPSIIA microglia. And we also see a lot of lysosomal genes that are upregulated, like CTSD, cathepsins, which are very important for the degradation of secondary aggregates and also glycosaminoglycans. So this brings me then to the part where we try to then leverage this information that we got from the observation of MPSIIA microglia. Can we use those to then make a more targeted therapy for Sanfilippo syndrome? And my lab focuses on 2 ways. One is the cell replacement therapy, what I talked first about, and then also trying to shift microglia. As I mentioned, they can be beneficial, they can be neurotoxic, towards a more beneficial phenotype. I start with the cell replacement therapy. So one idea would be to remove the diseased microglia that are not capable to degrade glycosaminoglycans, that are like over-flooding the brain with pro-inflammatory cytokines to get rid of them and then replace them with genetically corrected microglia. Similar to what's going on with the autologous hematopoietic stem cell therapy, but now more targeting microglia to get them directly into the brain. And one beneficial effect of replacing diseased microglia with genetically corrected microglia would be then to really enable microglia to deliver functional enzyme, in this case sulfamidase, to other cell types in the brain. So then also really to bring sulfamidase to neurons so that they remain functional. And this is a collaboration with Matthew Burton-Jones from UC Irvine, who really pioneered to replace diseased microglia with corrected microglia. So really the idea is to bring wild-type microglia back into the brain and then to remove the storage. So basically what we do is we generate corrected microglia or induced pluripotent stem cells, precursor cells, and then we transplant them into the brain of mice. So this is a preclinical model as well. So this is shown here, everything in green. These are human microglia transplanted into the brain of a mouse. So you can nicely see we have usually 6 injection sites. You can see here nicely that in the forebrain, within the cortex, and here's the cortex, here's the olfactory bulb, we have a replacement of microglia with human microglia. Not all places are targeted, so we're working on better targeting microglia and replacing them. Here's the cerebellum, which was not replaced by the corrected microglia. So what we did is we basically already very early on transplanted microglia into the brain of those mice, usually around day 2 after birth, and then we sacrificed the mice after 4 months. The first readout that we had was just looking at the glycosaminoglycan accumulation. So we have here the wild type. Usually on a wild type mice, you cannot detect glycosaminoglycans or non-reducing end products of heparan sulfate. In red, you have here now the MPSIII A mouse. You can see here you have an accumulation. In the control, just treated with PBS, we don't see a difference between both groups. But when we injected genetically corrected microglia into the brain of the MPSIII A mice, we saw a significant reduction in the glycosaminoglycans and non-reducing end products in those mice. We're also then checking, can we detect microglia, human microglia, still 4 months after the transplantation in those mice? And this is showing, again, single-cell RNA-seq, that we can detect still functional corrected microglia in the brain of those mice. Another project, the other approach is then really to restore, and we heard this in the previous talk, to restore microglia function, to reduce new inflammation, to boost lysosomal function in those mice. So this is a project together with Tony Whisk-Correa at Stanford. So Tony's group found that certain small molecule, C381, when you did a library screen, was really effective in reducing new inflammation and boosting lysosomal function, and actually reduced neurodegeneration in 2 mouse models. So we're looking at a Parkinson's disease mouse model, the MPTP mouse model, which has a very strong neurotoxic effect. And what I show here is a mouse model for neuronal ceroid lipofuscinosis. So it's a granulin knockout mouse model. It's also affecting granulin, the lysosomal function. And what you can see here, that in the granulin knockout mouse model, you have an accumulation of LAMP2, which is a lysosomal marker, and also CHIM2B. So you have increased lysosomal activation. When you treat those mice with C381, you have a reduction in the lysosomal activation and also in the immune inflammatory response. And this is another example. So again, EBA1 positive microglia in wild type and granulin knockout mice, plus minus treatment of C381. And here they were looking at galectin-3, which is a marker actually for microglia activation. And you can nicely see here that C381 reduces microglia activation, and also in this mouse model reduced the synaptic loss shown here. And so what we are working currently on is really, if this, together with Tony, if this component C381 is effective in the MPSIII A mouse model. So we have 3 groups together with the Scripps University. We are looking here at plasma neurofilament light. So this is a marker for axonal degeneration, which is also up in Sanfilippo Syndrome. We're looking at heparan sulfate and non-reducing end products. We're currently in the process of analyzing the immunohistochemical data. We're doing electron microscopy. And we have already some preliminary RNA-seq data that shows that this compound is anti-inflammatory in the mouse model. One drawback of the mouse model that I didn't show, the synaptic loss is very mild. So we are at the moment trying to figure out if we can better measure synaptic loss in this mouse model. So the current summary and future outlook are really, I hope I could convince you that microglia really play a very dominant role in the pathogenesis of Sanfilippo syndrome very early on in the course of the disease. And I think this could be really part of a multimodal approach to the treatment of Sanfilippo syndrome. Microglia become very reactive in Sanfilippo syndrome, and we think that we can twist microglia in a way that they can slow down the disease progression through shifting their phenotypes to a more beneficial state, so they are not detrimental. And another way is also to replace diseased microglia with genetically corrected microglia. My work is mainly focusing on MPSIII A. We started now a collaboration with the Characterisation lab, and especially with Raphael Grau here at UCSD to look at another mouse model of Sanfilippo syndrome. So we are starting in July a collaboration testing C381 for the treatment of MPS IIIC. And we are continuing to work, and I didn't show this work today, a little bit on the epigenetics of Sanfilippo syndrome, targeting a distinct transcription factor to enhance lysosomal biogenesis and to enhance lysosomal function. And this is ongoing work too. A lot of people beyond Chris Ballack are working on this, especially my mentor Chris Glass, who I had the opportunity to work with during my postdoc, and he was really supportive to let me actually go into the Sanfilippo syndrome research. And then collaborators, especially Jeff and Philip here, and at UC Irvine, Matt, and in Stanford, Tony Wisk-Gray. Thank you so much.
Thank you, Johannes. Are there any questions in the room or online? 1 second.
Hey, Johannes, that was a beautiful talk. Thank you very much. So did you do the single cell on your transplanted mice? Is that right?
That is right, yes.
Did you have a look at the neurons in that data set and whether they showed any improvement, if you're thinking that the healthy microglia can give some functional enzyme to the neurons?
So we are in the progress of looking at it. Hopefully in a week or two, or latest in a month, I can answer this question. So at the moment, I'm not very comfortable to answer the question. At the moment, I'm focusing, and we saw this yesterday, on this disbalance between excitatory neurons and inhibitory neurons. And I think we see a difference there. But I have not looked at if there's a cross reaction with the neurons regarding the enzyme.
Okay,
Yes.
no worries.
Very good
Looking
question.
forward to hearing. Yeah, looking forward to seeing whether that happens or not.
Yeah. I mean, there's a big discussion if really that's happening, this cross reaction. So yeah, will be interesting to find out.
Well, even if the glia are healthier and can provide more support to the unhealthy neurons, that would also be beneficial.
Right. So that's the hope, that actually the microglia then can really remove the heparan sulfate, the glycosaminoglycans, and so on. And this should make it into a more beneficial or healthier environment, and so that the neurons become more functional and don't die that fast. Because I do think that microglia actually determine a little bit how long a neuron lives. And we saw this yesterday also a little bit with the astrocytes and so on. We didn't see that many differences in astrocytes, but to be honest, with the 10x system, we don't get that many astrocytes. So the number of astrocytes that we captured was very low.
Right.
So
Single cell or single nuclei?
that was single nuclei, yeah.
Interesting. So you didn't get as many, there's a selection bias for nuclei?
Yeah, it's the 10x. So the platform that we use, it's a little bit, the astrocytes don't like the microfluidic system. I think a lot of them die during that process.
Okay, thanks.
Johan, I've actually got 2 questions. I'm wondering if you have any thoughts about the lifespan of your transplanted microglia.
So we looked up to 6 months. That's the longest time point that we have looked. I know that Matt, he has looked at later time points, and I think his longest time point was a year. So they survive for a very long time. Matt is doing fantastic stuff. He's also one idea is really to get rid of the diseased microglia. So the idea is there to use a selective compound that depletes microglia, and it's called Plexicon. So it's an antagonist for a survival factor of microglia, so CSF1R inhibitor. And I mean, we haven't done that experiment, but it would be a low-hanging fruit, I think, to see what's happening, at least in the mouse model. When we get rid of the diseased microglia, how does that affect the disease pathology? So if they become really detrimental at some point, it should be beneficial to just remove the microglia. And this compound is very effective in removing microglia. Has been studied in other disease contexts, not in Sanfilippo syndrome. So it has been studied, for example, in where we know there's a lot of microglia activation going on in multiple sclerosis. So there are clinical trials going on there. But we haven't looked, but that would be another way to look or find out more if microglia are beneficial or detrimental at some point during the disease course.
Just to follow up on that, removing the diseased microglia is essentially like a non-modified HSCT, isn't it?
Yep.
Which I think we in the field are fairly sure is not efficacious. So that's work done in both 3A and 3B mouse models from Brian Biggett's group and also from ours.
So I think
I'll be intrigued to see what you find.
I also think that just removing them won't be beneficial, but I think removing them and then replacing the diseased one with genetically corrected microglia, or also if you open up the niche, also if you inject then monocytes or macrophages into periphery, they have now a niche to then enter the brain. That also makes then, for example, stem cell transplantations, what is going on, a little bit more efficacious. So
It's that genetic correction, I think, that is the key factor. And that's certainly, you know, the work that Brian Bigger's done has shown that you can transplant mice with non-corrected cells, and there is no improvement in those mice compared to lentiviral modified cells that has progressed to human clinical trial. And we heard data yesterday from Simon Jones, and that's looking really promising.
Yeah, no, I think so too. Yeah.
Yeah. My second question was more around the nuances of the C381 treatment. I think it's a really intriguing approach. What you're trying to do is move the microglia, of course, to something that's less pro-inflammatory. I'm wondering long-term what you think, you know, if this is efficacious in doing that, long-term, how will you administer a drug that is designed to keep everything in a low inflammatory state when actually a move towards a pro-inflammatory state is a normal process, albeit it should be a short process to deal with the pathogen?
Yeah, I mean, I don't think that C381 alone would do it. So I think I see more C381 as a supplementary, or in addition to, for example, enzyme replacement and also the gene therapy and so on. The C381, also we don't know what it does actually on neurons. So I think I have to talk to Cedric also to really also test C381 in his brain in the DISH compound, because I have not the ability to look at neurons. I know that Tony with Correa, he is looking, so he has now actually founded a company in San Francisco looking at C381 for the treatment, not for Sanfilippo syndrome, but he was looking at NPC and also in lipofuscinosis, where they make actually a formula that's more oral, because this would be like IV treatment, but that is like also orally available C381. It crosses the blood-brain barrier, so it should engage with a target that is microglia, and then also hopefully with the neurons.
Might leave it there in the interest of time. If everyone can join me again in thanking Dr. Schletzky.
Thank you so much for giving me the opportunity. Yeah.
Our next speaker is Kai-Jing Zhu, who is a PhD candidate at the University of Adelaide. He's undertaking his PhD in the Alzheimer's Disease Genetics Laboratory under the supervision of Associate Professor Michael Idelli and Dr. Carissa Baltzan. During his PhD, Kaijing is using zebrafish and fly models of Sanfilippo to study Alzheimer's disease and Sanfilippo. Thank you.
Hi everyone, my name is Kaijing, a PhD candidate from Adelaide Uni. I'm going to talk about the Drosophila MPSIII mutation model for screening of potential therapeutic affecting lysosomal identification. The Sanfilippo syndrome is the childhood dementia and kind of the lysosome storage disorder. The metabolism, meaning there is the problem with the chemical reaction that naturally occur in the body. In this disorder, it's caused by a lacking one of the enzymes that normally break down and recycle a large complex molecule. In Sanfilippo, the carbon sulfate will accumulate to cause the lysosome deformation and enlargement. Here we use the fruit fly Drosophila to study Sanfilippo. We work with Drosophila because they are easily cultured in the lab and has the large number of progeny with the short generation time. Also, they are very genetic tractable, meaning that it can do very good genetic experiment and easy to look at the molecule change. Also, it has the highly conserved cell process and gene compared to human. There are 4 mutant fries pre-extinct and obtained from the Burlington Store Center. To determine which MPSIII mutation caused the severe phenotype, we measure the carbon sulfate concentration by mass spectrometer method. There are 3 samples in each genotype control, and each sample contains 5 6-week old homozygote flies, which rise in normal culturing condition at 25 degrees. The results show the SGS SCC4W has the most severe phenotype, and the heterozygote has the similar carbon sulfate concentration compared to the wild type. Before running the behavior assay, the mutant has the different genetic background compared to the Canton S wild type. So the mutant was outbreeding with the wild type ALS3 generation to exclude any genetic background effect. Each time when the cross were mating, the parent's male fly was collected and to do the PCR genotype and keep the heterozygote mutant flies. After the 3 generation outbreeding, increase the progeny to get the and genotype the both parents after mating, so we can get the outbreed homozygote mutant stock. When running the behavior assay, the outbreed homozygote stock flies will outcross the Canton S wild type, then increase to get the unknown genotype fly family for later experiment. After the experiment, the fly body will collect to do the PCR genotyping. We can get a quarter of homozygote wild type and heterozygote mutant and half of the heterozygote mutant. The advantages are there is less genetic background influence, and it must be blind for the later behavior assay. In the behavior assay, we want to know, does the mutant cause the reduced lifespan? The outbreed fly family was collected and rise in normal culturing condition at 25 degrees, recording the date and collecting the fly body for genotyping. The median survival of the homozygote mutant is significantly lower than the other genotypes, and mutant flies have the reduced lifespan compared to the non-mutant flies. And heterozygote mutant have the similar lifespan. As we can know, the motion defeat is the significant clinical trial in the Sanfilippo. We want to know, does the mutant fly affect the negative geotaxic behavior? In the Drosophila, they will uncomfortable move away from the ground. In the mass climbing assay, 25 flies will be measured over 25 seconds on the 500 milliliter cylinder at the same time. However, in our fly, they are unknown genotype until PCR genotyping after all assay completing. So I changed it to analyze individual flies with the genotyping determination later. This tube has the same height but thinner than the cylinder. If the fly cannot complete the 300 mark, it means climbing defeat. If it less the 50 mark, means it lost the climbing ability. We can get the tracking imagery from the ImageJ analysis. The results show the mutant fly have a more severe decline in climbing ability over time. We made the day 28 is the first day, so we can see the mutant fly have the reduced climbing ability compared to the non-mutant flies, and the heterozygote mutant have the similar climbing ability compared to the non-mutant flies. In addition to the climbing, the vertical moving, we want to know the does the mutant cause the spontaneous activity. The Drosophila activity monitor system is the method to measure the spontaneous activity in flies. When the fly across to the infrared beam to eat food, one active motion will be recorded. Here I use the 16 days old and rise in normal culturing condition outbreak fly family. The spontaneous activity was recorded continue under normal culturing condition for 7 days on a 12-hour dark light cycle. The results show the mutant will affect the spontaneous activity. The mutant spend less time to alter spontaneous activity than the non-mutant fly, and the heterozygote mutant fly have the similar activity compared to the non-mutant flies. The lysosome acidification is a necessary pathway to maintain enzyme activity. The lysosome acidification is a key feature to healthy cells, and that fail to maintain pH is linkage to aging and neuron degeneration. Many degeneration diseases have been shown to be associated with defect in the lysosome acidification, like Alzheimer's disease, Parkinson's disease, and MPS I. So we want to know is the lysosome acidification defect in the Sanfilippo. For lysosome acidification determination, we decide to fluorescent fusion protein gene linkage to endogenesis lysosome protein LAMP1, which is the highly conserved, and its overexpression will not affect the lysosome function. The pH fluorescence is the pH-dependent protein. It will provide the green fluorescence when pH more than 5. And there is the pH-independent protein M-cherry. It will provide the fluorescence over time. Also, we have another pathway to determine lysosome pH. The lysosome pH determinant agent red is the red fluorescent dye that will accumulate in acidic lysosome, and its fluorescent intensity increased as the pH increased. Also, the LAMP1 GFP1, the LAMP1 GFP is the fluorescent fusion marker for co-localizing the lysosome. Also, we are testing 5 potential organisms to rate acidification on the mutant flies. Thank you very much for listening. I would like to say thank you to Professor Michael and Dr. Carissa for everything they have done for me. Of course, the Sanfilippo Children Foundation support.
Thank you. Does anyone have any questions for Kai-Jing?
Do you know if there's markers of ER stress that are able to be employed in Drosophila?
What do you mean, the ERs?
For ER stress?
No, I don't know.
Sure, okay. Yeah, something maybe we can talk about afterwards. That's just something that I'll speak about a little bit later on, about the potential for residual protein to initiate an unfolded protein response in ER stress.
Yeah.
But great talk, Kai-Jing.
Okay.
Can I just clarify? You looked at 2 different fly lines? Is that correct? 2 Sanfilippo fly lines?
For the mutant fly is the
When you're doing
same.
your heparin sulfate, was it? Yeah, oh, sorry.
The other one is white type, one is the mutant, and the heterozygote mutant. Heterozygote mutant should be similar to the white type, but we don't know yet, so we're just testing at the same time.
Beautiful. Thank you very much. Everyone can help me thank Kai-Jing again?
Yeah.
Great.
All right, we might make a start. This session is entitled Modelling Sanfilippo Disease and Symptoms. I'm standing in for Cedric Barty, who unfortunately is unwell and unable to join us and share today. My name's Kim. I've worked along with my colleagues in the Childhood Dementia Research Group for around about 20 years. We're interested in Sanfilippo syndrome predominantly, but we're also branching out into other lysosomal storage disorders. We work on cells, on mice, and increasingly now on other model systems, including flies and fish. And most of our work is directed to finding therapies. Our first speaker today is Ewan Gerken. Ewan is a PhD candidate at the University of Adelaide. He's a recipient of a Sanfilippo Children's Foundation PhD top-up scholarship. He's undertaking his PhD in the lab of Associate Professor Michael Lardelli, and he is co-supervised by Carissa Barthelson. He uses zebrafish models of Sanfilippo, but also early onset Alzheimer's disease to study how those diseases are similar and different and how they affect brain cell and zebrafish behaviour. Thanks, Ewan.
Thanks, Kim, for that nice introduction. So yeah, as she said, my name's Ewan, PhD candidate at the University of Adelaide. And today I'll just talk you through how we came to get these models generated and some of the work that we've done on them so far. So firstly, a bit of background. Now, Carissa did answer some of this before, so I'll sort of gloss over it slightly. Traditionally, our lab has been focused on using the zebrafish model to investigate the genetics of familial Alzheimer's disease. We like this model for several reasons. The zebrafish, as Carissa said before, share similar brain architecture to humans, and the brain neurochemistry is pretty much conserved as well. They also share a wide range of disease-associated genes with humans, so about 70% orthologs in zebrafish, and that includes a lot of genes linked to neurodegenerative diseases such as Alzheimer's and also a lot of MPS disorders. The real advantages of zebrafish, which really set them apart for us, however, lie in their reproductive characteristics. So as we went through before, a single mating event between zebrafish can result in up to 100 embryos per mating event, which develop extremely rapidly with a low cost of care. So this allows us to test greater numbers of subjects within single families, reducing the effect of genetic and environmental factors on the results. So as Carissa said, basically they're cheaper, easier, faster than more traditional models such as mice. So we decided to seek funding to apply these advantages to childhood dementia disorders. So the Sanfilippo Children's Foundation were kind enough to provide us with some funding to create zebrafish models of MPS IIIA, B, and C. And these models we've created, 3A is the second zebrafish model after Jan Kaslin's model. Sadly, I don't think we'll be able to hear about that today, but they've done a lot of good work with that. But we have the first zebrafish models of MPS III type B and C. The gene GNS responsible for type D is duplicated in zebrafish. This complicates analysis, so that's why we excluded this gene from our study. So shown here are the exon structures for the zebrafish SGSH, NAGLU, and HGS-NAT genes. Our lab's general philosophy for mutant models is to replicate the genetic state of the disease as closely as possible, as this relies on the fewest assumptions. So using CRISPR-mediated mutagenesis, we target our mutations upstream of known human pathogenic variants. So the idea behind this is that they were therefore likely to be equally as severe as their human equivalents. So we also chose mutation sites so that any introduced premature termination codons would be present only in the final exon. And this has the benefit of avoiding nonsense-mediated decay and preventing sort of any transcriptional adaptation associated with that, which can complicate analysis. So the mutants themselves, what did we end up with? So shown here are the aligned Sanger sequencing chromatograms. The human and zebrafish wild type sequence is shown on the top two there. And then our mutant sequence in the zebrafish is on the bottom. So for SGSH, we selected this 16 base pair deletion, resulting in a downstream frame shift. In NAGLU, we selected a similar 10 base pair deletion, and this also resulted in a frame shift. And finally, in HGS-NAT, we selected this larger 37 base pair deletion across the end of exon 17. So from these Sanger sequencing results, we can see that the mutations we've introduced significantly disrupt the amino acid structure, at least, of the carboxy terminus of each protein. Next, we sent samples of 3-month-old brains for analysis of heparan sulfate accumulation. So we found that heparan sulfate concentration remains at basal levels in wild type and heterozygous fish in all 3 mutants, and accumulates significantly in homozygous fish for all the mutants as well. This is nice to see because it means that our mutations are knocking down the enzyme function as they were designed to, and recreating the primary disease signature is obviously quite important for their use as a model. But what's the actual effect on enzyme function, and what's the level of this effect? So to quantify this, I performed a fluorometric enzyme activity assay with the help of people at Kim's lab at Flinders. And we did this on 16-month-old brain samples from wild-type, heterozygous, and homozygous fish from the SGSH mutant family. And we can see a nice dose-dependent relationship between consumed substrate and genotype, in line with what we would expect. However, somewhat surprisingly, it does appear from this data, at least, that the homozygous fish for the SGSH mutation do retain some level of enzyme function. So this is quite interesting. Due to the significant heterogeneity we see in the human disease presentation, we sort of think that our model may actually be beneficial in understanding attenuated cases because of this result. So I haven't yet performed enzyme activity assays on the other 2 mutants, but that is right at the top of the list to get done at some point when I have time. So it'd be interesting to get the results for those and compare to see, which mutation may be more severe in terms of the effect on enzyme function. All right, so moving to some cool behavioral work. As part of the grant from the Sanfilippo Children's Foundation, we want to see whether these mutations have any effect on the fish behavior as they age. So for each of our mutants, we create families of at least 100 fish by heterozygous incross for behavioral analysis. And this strategy is pretty similar to what Kai-Jing mentioned and Carissa mentioned before. The difference in this one is that we do a heterozygous incross compared to Carissa's heterozygous times homozygous incross. So the advantage of these crosses, again, is that they enable internally controlled intra-family analysis between genotypes originating from the same parent pair. And like Carissa said, we genotype after data has been collected. It's a good habit because it blinds us to the results, but also it stops the stress that genotyping puts on the fish from influencing the experiment. So to test our fish, we've done a lot of work using the Xantics free movement pattern Y-maze to assess a form of memory, spatial working memory, the same test that Carissa went through before. I'll give you a little bit more detail about how the test works. So the concept is that intrinsic curiosity will drive fish movement as they move through the Y-shaped tank for 1 hour. So when they reach the center of the maze, they face a choice of left or right. And basically what we do is we record the sequence of these turn choices. We then calculate the frequencies of each possible tetragram. And in this case, a tetragram is just a name for 4 consecutive turn choices. So it's been shown via previous work from the UK that fish, along with mice and humans, display a conserved natural dominance of alternation-based search strategies in the Y-maze, which you can see in this figure on the left. So the alternation tetragrams are highly overrepresented. And the assay has been validated as well as a test of working memory by data showing that zebrafish use fewer alternations when their working memory is chemically inhibited. And you can see that here. So I tested our behavioral families in the Y-maze for this experiment. We've done many ages, but for this experiment, this was 18 months, so a little bit older. So what we can see here are generalized linear mixed effect model predicted alternation probabilities for our 3 mutant lines. So in this experiment, we don't see any significant effect of genotype on the spatial working memory output for each of these models. So although there's no formal evidence to suggest that spatial working memory is affected in this experiment, I will point out that for the NAGLU family, the homozygous fish do show, although it's statistically insignificant, they do show a trend towards lower spatial working memory indications. So that's quite consistent with what Carissa was seeing. And it just may be due to a lack of power in this experiment. Carissa tends to get more power with the heterozygous cross homozygous families because you can get greater numbers, half, 50 of each for 100 fish family is a lot better than 25, 50, 25. Next, I looked at the predicted average number of turns performed by the fish during the test as a measure of general activity. So I found significant effects of genotype on predicted turns for SGSH on the left and NAGLU mutants. So the homozygous fish in each of these cases showed a higher turn rate. The difference in the HGS-NAT fish was not significant. However, it does appear there may be a similar trend there. We may just be underpowered in that. So there's evidence here of hyperactivity presenting at later ages in our models, which is consistent, not in terms of the lateness, but consistent in the hyperactivity observed in children with MPS III and some mouse models, although there's some inconsistencies out there with those. So I've done quite a lot of work with the Y maze. However, we also wanted to try a different test on our fish, just to see if we can get some different results. So what I've done is perform the novel tank diving test. The premise of this test is that the fish will naturally hide at the bottom of a novel or new environment and gradually increase their vertical displacement as they habituate to the environment. So you can see that this fish is habituating a bit faster than the other fish. So what we do is we define 3 zones within the test arena. There you go. And then we can measure outputs such as distance traveled, entries into, and time spent in each zone. And so this test gives information on anxiety phenotypes, which are common in Sanfilippo. So basically the idea is that less activity in higher zones is indicative of higher anxiety levels. So I've collected all the data for this experiment. The analysis is still ongoing. I have one very preliminary figure to show you, which is just showing the number of entries into the top zone of the novel tank performed by HGS-NAT zebrafish. So I haven't performed any statistical testing of these yet, but you can see on the right that homozygous zebrafish show a lower average number of turn entries into that top zone compared to wild type and heterozygous siblings, suggesting that anxiety may be increased in these fish. So there's still a lot of work to do with this data, obviously, but that's a little taste. It's important to note that the novel tank is adaptable for zebrafish larvae as well. So this data is collected at 6 months of age, so adults. But if we can detect robust changes in adults using this assay, we can then maybe look to move into detect similar changes in much younger fish. And those behavioral changes in younger fish are more useful ultimately for drug screening and testing treatments. Okay, moving on from behavior to transcriptomics. A large focus of the grant was directed towards performing an RNA-seq experiment in the brains of our MPS III zebrafish. And most of this analysis was performed by the RNA-seq whiz, Carissa. So our strategy, again, a similar strategy, was to in-cross fish, this time heterozygous for 2 of our MPS III mutations. So in this way, we get a large family, which we raise together, same as before, but we get a greater multitude of genotypes here. But the ones of interest are the wild type fish, homozygous fish for 1 mutation, and then homozygous fish for a separate mutation. So we repeated this family setup for each pair of mutations, resulting in 3 families, each containing a 3-way comparison. So this is really nice. The advantage is that it enables direct intrafamily comparison between Sanfilippo subtypes, and we get inherent replication in comparing each homozygote to wild type fish twice. So it's a really nice setup, at least in theory. So first, gene set enrichment analysis was performed to give us a list of significantly altered KEGG pathways. What I'd like to point out here, just broadly, is that the AB and BC comparison arms of the experiment, the middle column and the right-hand column, appear to be underpowered, as we detected many significantly altered pathways on the left in the AC arm, but only the most powerful changes in the AB arm and none in the BC arm. So it's more than likely that this can be attributed to some difficulties we had with the family structure for 2 of these arms. So there's some limitations to this result, but I'll take you through what we've been able to find out. So we have a look a bit closer. So reassuringly, we detected strong changes to lysosome and glycan degradation gene sets in all 3 mutants. We also detected significant changes to pathways involved in things like sugar and lipid metabolism, as well as inflammation, which is also consistent with what we know about the disease. We also looked at whether cell type marker genes were differentially expressed in the MPS III zebrafish brains to give us an insight into whether our mutations might be having a stronger effect on some cell types over others. So we found significant changes to neural stem cell gene sets in type A and C fish in the most highly powered arm of the experiment on the left there. Really interestingly, we saw changes to oligodendrocyte gene expression in the brains of all 3 mutants, suggesting that this cell type may be strongly affected by heparan sulfate accumulation compared to other cell types. This would actually be quite biologically intuitive, as oligodendrocytes have a really large surface area and would theoretically be under a high demand for extracellular matrix and membrane turnover. So that's quite cool. And this data is actually also supported by recent work, really nice recent work from another group using a 3C mouse model. Finally, we performed an enrichment analysis on our iron-responsive element gene sets. So these gene sets are simply a list of genes which contain elements contain elements in their untranslated regions, which respond to changes in iron supply. So we found strong evidence of changes to these gene sets in MPS IIIA zebrafish in particular, and we weakly detected some changes in MPS IIIB fish as well. It's likely that those weaker changes are due to the lower power of that arm. In fact, that arm there, where you see the change to the MPS IIIB fish, that was probably the most underpowered arm of the experiment. So the fact that we see something still is quite encouraging for the Naglu fish. And again, we detected no changes to iron homeostasis in the 3C fish, although, yeah, there were some limitations to the power of this experiment. So yeah, this ties into our interest in the role of iron that Carissa went through earlier, to Sanfilippo and other neurodegenerative disorders. Okay, so that's about it. In summary, MPS III model fish appear to show phenotypes reminiscent of the human disease, particularly that primary disease signature of heparan sulfate accumulation. We've also seen that our MPS IIIA fish show some enzyme activity retention, which may be useful for modeling attenuated disease phenotypes. Hyperactivity phenotypes have been detected at later ages, which is encouraging because we see this in humans with Sanfilippo. But the question is, can we detect these types of changes earlier to make them more useful for further work? We also see brain transcriptome changes, which are consistent with our understanding of MPS III in humans, as well as new evidence of changes to oligodendrocytes and iron homeostasis, which are potential targets for follow-up experiments. So further investigations into these models are definitely warranted, but we believe they can be a really important, useful tool in better understanding Sanfilippo syndrome, with the eventual goal of identifying and testing disease-altering therapies. So as you see a nice QR code there. Most of this work, if you're interested in learning a bit more, is available as a preprint on bioRxiv, so feel free to check it out. I'd like to thank everyone who's contributed to this project. There's been quite a few people, all the other lab members from the zebrafish and Drosophila facilities. Thanks also to the Cartu Foundation for their continued support over the lab over the years. We probably wouldn't exist without them. The biggest thank you, however, must go to the Sanfilippo Children's Foundation, not only for supporting me throughout my PhD with a top-up scholarship, but also their funding contribution towards this work was instrumental, and for their efforts in putting really important events like this together, where we can bring in clinicians, basic science researchers and families to sort of, come together and keep working towards finding solutions to this disease. Thank you.
Thanks very much, Ewan. Do we have any questions in the room? Maybe Al? Yeah.
With your hypoactivity data in your fish, have you looked at older ages? Sorry, hyperactivity data. Have you looked at older ages to see whether you've got a hypoactive phenotype?
So those fish were 18 months, which is fairly older than we usually would do testing at. Basically, there's a whole sort of backstory here, which I didn't have time to go through, but when I started doing behavior on these fish using the Y maze, we had the machine we had to do the testing had a capacity of 2 fish per hour, and we had 1 machine. So basically testing a whole family would take place over the course of a week or 2 weeks really, all day, every day. But there's just so much variation doing something like that. So a lot of the data that came out of that was really unclear. And even in this data, you can see that there's so much variation between the individual fish. So the data I showed was after what we did after we basically increased our testing capacity. So we can do like 16 fish at a time. So it's increasing the throughput, but also making the data a bit nicer, a bit cleaner. So that's why they're older. They're the same families that were tested at younger ages. We didn't detect anything at younger ages, and so we found some hyperactivity at the older ages. We haven't gone further than this. And I know what you're going towards, because hyperactivity is usually seen a bit younger, then it falls away as the patients regress. You know, it's possible that for the zebrafish, everything is just shifted back in terms of the time frame, because, you know, another difference to humans, they're quite regenerative, so their brain can quite strongly resist maybe like gross neuronal loss that you might see in humans. So that sort of stuff might take longer, but we haven't gone further than this age, yeah.
And sorry, what's the lifespan again for a zebrafish?
In the wild, 2 years. In the lab, they can go much longer. We tend not to keep them for long after 2 years. Yeah, if you look up, they can survive up to 4 years, 4 or 5, but, you know, ethically, we tend to not let them go that far.
Maybe if I can ask a question. You started by outlining the conservation of genes associated with Sanfilippo syndrome, and you know, 3A to 3C are there. 3D is complicated, but I wonder about the gene that has been associated with MPSIII, or maybe Usher syndrome, which is an arosulfatase gene. You looked at that one?
I'm not sure if that has an ortholog in fish. Yeah, no clue. I mean, you know, limited resources, we try to focus on, and, you know, the thing with 3D as well is it'd be nice to study, but it was just too complicated and also so rare. And then E, from my understanding, is rarer still. So yeah, we've just focused on A, B, and C. I'm not sure about E.
Right. Now we're going to move along to the last talk in this session, because as Ewan mentioned, Alon is unfortunately unwell today and couldn't join us. You've heard about me, so I think I can just launch straight into the talk. But essentially what I'm going to try and do here is stimulate a discussion that will follow. So please be thinking about questions as we're moving through. And the answers we're seeking is, do you think that we need more models? Have we got enough models to understand these diseases? Have we got enough models to test potential therapies in? And if you think the answer to that is no, then please come up with what you would like to see in a model system. And I'm interested in the contributions from families, from researchers, from our patient societies, and also from clinicians. All right, I'm going to run through what we've got. So it's going to be a whistle-stop tour because there's actually a lot. I'll start with dogs. There's 2 dog colonies in existence for Sanfilippo's type A and B. I'm not going to talk about the dachshund that has MPSIII A, purely because by my definition of a model system, I'm limiting the discussion somewhat to models that have been used beyond a description of what they look like, where people have tried to gather more information about diseases and test therapies. So what you can see in that table is that we're missing some models in dogs. It doesn't mean they don't exist. It just means they haven't been reported. These models in dogs are naturally occurring. People haven't created these. They just occur like they do in people. So here we're going back to work done in the late 90s and early 2000s. And we know about these dogs, and the models have been established purely because we have amazing veterinarians out there who themselves are also curious and scientifically minded beyond their veterinary work. And they come across these animals in their clinic, or the animals are referred to them, and their curiosity gets the better of them. They believe it's a metabolic disease and they seek out collaborators in order to get a diagnosis. So Bob Jolly, Emeritus Professor Bob Jolly in New Zealand, Massey University, led the diagnosis of the Hunterway dog type. And that dog, as I'll show you in a moment, has had a massive contribution to the Sanfilippo field. Similarly, Matthew Ellenwood and colleagues, initially at UPenn, diagnosed and set up colonies and have utilised, again, to very significant effect, a Schipperke model of the Sanfilippo type B. Very briefly, I'll just talk about one paper, and this is really work that came and is part of a very, very large body of work that started in mice and moved into dogs. But this is just one study, and I'm sorry, the words there are very, very tiny. Essentially what it's doing is summarising one study where we put replacement recombinant human sulfamidase into the cerebrospinal fluid in those dogs on a repeated basis to see what it would do to the brain. We knew from our mice that this was likely to reduce substrate burden, and it was very likely to reduce the neuroinflammatory effects of the disease. And indeed it did, to beautiful effect in those dogs. Those studies were short-term. The enzyme is very expensive to make, and dogs are very expensive to maintain. So short-term studies, unfortunately. But it did lead to clinical trials, a number of clinical trials spread across the world in patients with Sanfilippo syndrome. These studies showed that the treatment was by and large safe, reduced the heparan sulfate load presumptively in brain because the heparan sulfate load went down in the CSF. You can see that in that top graph. But the patients that are at least reflected in the graphs I've got here in front of you today, the patients in these trials, because we don't have newborn screening, are patients who are often well into their disease journey. And in this part of the study, the mean age of entry into that study was 10 years of age. And what you can see in the bottom graph is that, sadly, the patients continued to decline despite the presence of repeated doses of sulfamidase. That trial has subsequently been halted. This is work that is more recent and was carried out by Matthew Ellenwood and a large team of collaborators in the 3B dogs. They're essentially doing the same thing, but with a slightly modified form of the NAGLU enzyme. The enzyme's being put into the cerebrospinal fluid of the dog, again, on a more frequent and higher dose basis. And what has happened here is they've actually extended these studies out a lot. So they've gone out to 2 years, which is a much more informative experimental series. And they've also undertaken some behavioural studies in the dogs. So the graph on the left there shows that compared to untreated and then affected dogs, the 2 bars on the right have the treated dogs acting more like the unaffected dogs. So that's very promising. And it led to establishment of clinical trials of intracerebroventricular replacement enzyme in patients. And again, this data is very positive. These children were younger when they went into the trial, approximately 5 years of age. The treatments are more regular, higher dose, but I think the data is potentially suggesting that there is at least some stabilisation there with the treatment. So to just briefly summarise that, the benefits versus the costs. There's some obvious benefits. The studies have led to human clinical trials. And this is the models have been used beyond mice because we're increasing the size of the brain when we move from a mouse to a dog. The brain is far more complex, and it's also going to give us a better indicator that if we put a treatment into a specific region of brain, is it able to move around the brain and affect treatment across a very large area, which we're hoping it will do in a patient. And this is in contrast maybe to an IV delivered or an oral delivered treatment where we might expect it to have more global access. This is balanced against an ethical cost. I can tell you from my perspective, working with the dogs, and I wasn't even hands-on with them, just at the post-mortem and laboratory end, there's a very significant ethical cost. There's also a highly significant financial cost. They require lots of room for housing so that they have a nice life, long generation time, and also you need very highly skilled people working with them. Of course, veterinary care is essential. So where are the dogs at the moment? The 3A dogs are no longer in New Zealand. That colony was shut down. Funding is essential to keep those colonies going, but the sperm was frozen and it's been shipped to the US. The 3B dogs are in existence in 2 places, in both the US and in France, and they're under active study. To the best of my understanding, there's no dog models of MPS3 C, D or E. And as I mentioned earlier, they're probably out there, but probably increasingly rare, and it requires somebody to notice them and get them identified. But I want to end here, and I'd like to come back to, in the discussion, a bit of a thought process amongst the group and online about whether we value these models enough to keep them going. Because I can tell you from discussions with Matthew Ellenwood and others in the field that the colonies are absolutely precarious. It's incredibly difficult to keep them going without a large financial contribution. So we as a field have to have a discussion about whether we want them, and if so, how do we fund them and keep them going? One of the reasons we want to keep them going is because they can really inform us about what you do in a patient. And this is an example of this in CLN2 disease, Batten disease. This is the Brineura or Silipinase alpha, and the data that came from CLN2 dogs and also some unaffected monkeys was used to scale up the dose that was used in patients to great effect. All right, moving on. Now you probably didn't think you'd hear about goats this morning, but I'm going to do a brief stop with goats. The 3D goat was identified, I think it was actually in about 1992, but there's some cloning and sequencing that was done a little bit later. Margaret Jones, who I understand was is a veterinarian, or was a veterinarian, is a person, and obviously. I've worked with these goats and studied their disease, but importantly, undertook a very early enzyme replacement therapy trial in the goats. At this point in time, we didn't fully understand whether replacement enzyme, if it was delivered intravenously, would access the brain or not. And there were questions around this because at the same time, MPS6 enzyme replacement therapy was ongoing in MPS6 cats. And this is a treatment that's now in use in MPS6 patients. So it was effective in MPS6, which does not predominantly affect brain, affects bones and joints and other parts of the body. So the question here was, can it get into brain? And so this enzyme was synthesized in the lab, which is an expensive undertaking, but there was enough enzyme that was made for a couple of doses in a goat. But as we now know, the outcome was it does not reach the brain. So informative and very early study. Moving on to mice. There are a number of mouse models in existence, 7 to my count at the moment. I hope I haven't missed anybody's model. 7 for MPS3A and 2 of each for 3B and 3C and 1 of each for D and E. I'm going to go through some of these, but not all of these, to briefly pick out the alpha-synuclein cross mouse that we made some years ago. Crossing Sanfilippo type A mice with a D31N mutation, at least with an alpha-synuclein knockout, made no difference whatsoever, telling us that very simplistically, with the markers we looked at, alpha-synuclein doesn't play a major role in the progression of Sanfilippo type A. And then I'll go through most of the others of those, I think. I don't think I'll touch on D and E. There hasn't been quite as much work done on them, but there is an evaluation of therapies in MPS 3D. Now, Matthew Ellenwood undertook a really interesting experiment in both the 3A and the 3B mice, the first models that people either found or created. And he crossed those mice with a knockout mouse line that meant that the Sanfilippo mice were not able to make GM2, which we know accumulates for reasons that are still not fully delineated. It accumulates in Sanfilippo and other lysosomal storage disorders. But the idea here was that if that GM2 is driving the disease process, if you get rid of it, maybe that might actually slow the disease. And another factor behind it was that there was a therapy available to inhibit GM2 synthesis that is available for other disorders. So Matthew crossed these mice and crossed his fingers and hoped for the best with the prediction that this would hopefully make the disease better. But as science often shows you, you don't get what you wish for. The mice actually fell off the rotor rod sooner and they passed away sooner without that GM2 ganglioside. The hypothesis was that it was some of the other gangliosides that were present that were actually toxic. And side by side with that, the substrate inhibition drug miglustat had no effect in a trial in patients. Lexi Pashetsky's done some beautiful work creating and evaluating and utilising Sanfilippo type C mouse models. He created the mouse knockout, the very first mouse, and more recently a mouse that expresses a misfolded sulfamidase protein. So this one has some residual enzyme. So here, I think what he was predicting was that if you have a little bit of enzyme activity, you're going to be better off than if you don't. When you looked at heparan sulfate levels, they were no different between the two mouse types, and maybe that is because there's just not enough HGS-NAT there. But the deeper he delved, the more he found that the knockout and the missense mutant are not the same, and the missense mutant actually seemed to have a more severe phenotype. A number of behavioural studies, as well as a lifetime analysis, showed that the HGS-NAT missense mutation mice not only had a worse phenotype, but they died earlier. They also had a more progressive neuroinflammatory response, and they accumulated ubiquitinated proteins. Alexei hypothesizes that this is potentially due to an unfolded protein response and ER stress, and that you've actually got toxicity caused by that mutant lysosomal enzyme that's being produced. We've done a similar study and compared the D31N MPSIII A mouse model that is a missense mutation and some of the time has some residual enzyme in our enzyme activity assays, and we would predict that the knockout would be worse than the residual enzyme mouse. In a head-to-head comparison, at an age younger than we would normally see a phenotype in Sanfilippo missense mice, we saw a phenotype in knockout mice. So the knockout mouse did seem to have an earlier onset phenotype. But other than that, there was no difference. So that difference in the phenotype couldn't be explained by heparan sulfate or any of the other disease markers, including ubiquitinated protein accumulation in those animal models. So those changes histologically, biochemically, could not be correlated with the phenotype. I think putting all of that together, the 3C and the 3A study is just another indicator that genotype and phenotype are very difficult to correlate in Sanfilippo syndrome. And maybe we need more models. What did come about as a result of this study, though, was a mouse model that's sort of sitting there in the background, and that's a conditional mouse model that can be crossed with a Cre recombinase strain. And it has the capacity, if people are interested, to create cell-autonomous models. And what I mean by this is if you want to understand the contribution of a particular cellular pathway, you depending on the availability of an appropriate Cre line, you have the capacity to make a disease model that's exhibited in certain cell types and ask, are they important? With relevance to cell replacement therapies, there are immunodeficient mice available for Sanfilippo type A. These mice are the D31N mice that are also knocked out for a couple of other genes that essentially gets rid of some of their immune system. They have reduced sulfamidase activity, as we'd expect. They look like they have a motor phenotype, and they're missing a lot of the immune cells or immune response, and this makes them amenable to human cell transplantation. So they're not going to have an immune response to those foreign cells. Interesting model. And finally, we're currently in the process of creating a model of the R245H variant. And this is one of the most common genetic changes in Sanfilippo type A in Australia. It's also quite common in the Netherlands and the US. It's associated with an early and rapid disease process. This is funded by the SCF and Foundation Sanfilippo Suisse. SA Genome Editing have created the mouse. Adeline has established a colony and a PCR method. She's now got a large phenotyping cohort on the ground, and the phenotyping will occur later in the year. The goal here is to characterise them and then put them out into the field so that everybody can use them. Adeline's data so far is that they're deficient in sulfamidase and they store heparan sulfate, which is good. All right, Drosophila. We've heard a little bit about Drosophila this morning, so I won't spend too much time on any of these things. Drosophila, I think they're amazing. They have heparan sulfate stored. You can see that in the top right. As a result of a change in the sulfamidase gene expression, and they can't climb up that measuring cylinder as well. But the unique thing about Drosophila is that you can change the expression of other genes and ask whether that hastens the disease or slows the disease down, and that can be really helpful for us in understanding where we need to go with therapies. So what Danny Weber, Louisa Keith and others showed was that manipulating heparan sulfate biosynthesis, whilst it sounds like a great idea to knock that down, to put less substrate into the system so that the sulfamidase that might be there has less to cope with, that has to be done with care, because if you knock it down too far, you actually get a worse phenotype in those flies. Fewer of them can climb. And the other thing they showed was that if you modify the way in which cells and organs deal with antioxidants, you need to be careful there as well, because you may actually worsen the phenotype if you overexpress some of the enzymes that are supposed to be dealing with antioxidants and free radical production. 3 C Drosophilas were created, and the paper was published at the end of last year. Again, they store heparan sulfate, they have this climbing defect, and what Laura also showed, a PhD student, was the flies have some problems with their sleep. Laura's also created some lines to start to unravel which are the important cell types for creating a phenotype. And so she knocked out the HGS-NAT in specific glial cell types and showed that there were a couple of them that actually worsened the phenotype, indicating that maybe they're cells that we really need to think about ensuring that they get a therapeutic because they're important for a particular phenotype. Zebrafish. I won't spend too long here because we've had some beautiful talks already. Alon and Jan's work is in the literature so far, but zebrafish models of 3A, B and C have been created. They seem to have some phenotypes, and I think they're going to be of great use going forward in understanding disease and also evaluating therapies. So this is the work Ewan's just gone through. And then finally, finishing up with cells, skin fibroblasts from patients are of great benefit to us in the field, and they can be turned into iPSCs, or they can be directly differentiated to other cell types. And so I'm not going to go into this because we had some beautiful talks yesterday, but these are very useful for drug screening, of course, and one of the exciting things is how high throughput you can be with applying drugs to them and asking if a particular compound is efficacious. So now, coming to the gaps, and I hope you've got some thoughts in your head about do we need more models and gaps? For my money, I think potentially we need some more knock-in mutations so that we can evaluate pharmacological chaperones, which are not going to be efficacious if you pop them in a knockout model. They need some residual protein to chaperone through. And I think they're also going to be helpful to let us understand the importance or not of misfolded protein. But they only exist for a couple of different diseases in mice and the flies that Kai-Jing showed us this morning with 3A. We are also missing models that the disease is only expressed in certain cell types that might help us understand the disease process better. We are missing attenuated disease models. So these are the still devastating, but more slowly progressing disease models. City Mobaroka is creating some neural cell models of attenuated Sanfilippo at the moment. It's a very new project, so not sharing the data today, but next year we plan to. What I want you to think hard about, because we've had a number of talks yesterday and today where AI is being used. And I'm wondering whether we have so much data in the literature on Sanfilippo, whether we can actually harvest that data, build upon that data and use it to predict potential therapeutics and also the pathogenicity of mutations. And I give you those two scenarios because there are people already doing that in the lysosomal diseases field. There's a couple of papers in the last, well, actually mostly this year, that are using AI for precisely those processes. And then finally, I just want you to think about worms. C. elegans is, I think, a really nice model system that we don't have for Sanfilippo, and we're behind the game on this. I don't know very much about C. elegans, but I do know a fantastic C. elegans researcher, Dr. Yi-Lian Chu down at Flinders. And from everything Yi-Lian and her group show us, it's a very versatile model system. They have a very short generation time, a bit like flies and fish. You can create lots of them, reduced ethical issues. They have an incredibly characterized nervous system. It's only got 302 neurons, so it's quite well understood and quite easy to study. The connectome is known, so how everybody talks to each other is known. It's amenable to genetic screens and I believe probably chemical screens as well. And importantly, the lysosomal genes, and this is actually an old paper now, so if people are interested, follow up on it, but I think the lysosomal genes are going to be conserved, and there's at least a couple that were already conserved when this was published. As I say, we're behind here. People in the Niemann Pick C, ML4 and the Batten disease fields have been exploring the value of worms for genetic and chemical screening. So in summary, we've got tons of them, but we are still missing some critical models. We've got some neglected subtypes, and if we want to be as giving to the field as possible, we need to actually explore having additional subtype models and more of them, not just one. We also have neglected disease presentations. We have a good representation of rapidly progressing disease, but not a more slowly, but equally devastating form of disease. So what I'm challenging you to do today is to think about what you think is the best or fastest pathway to approved treatments with the lowest cost. Cost shouldn't be a factor because every child's life is critical, of course. But as we heard yesterday from Simon Jones, cost of the trials are prohibitive, and cost of rolling out, say, a gene therapy into a population that we hope is all over the world one day, not just in wealthy parts of the world. We hope that we can come up with efficacious treatments that everybody can have. I think it's likely it's going to involve a number of models. So my question is, can we identify new drug targets faster and cheaper? And maybe we need to look at those non-traditional models. So things like the fish, the flies, the worms, and AI. And maybe those can funnel into some of the more expensive systems when you have some, really nice targets. But that's just my 2 cents. So over to you.
Very insightful and very comprehensive information. It's really good. So, so actually my not comment, not question, it's a bit contradictory to the challenge that you give, with the low cost and fast. So I wonder if you can comment on model of primate. Some drugs works really well in other neurodegenerative disease, neuroactive drugs, but they can only bind to receptors that is expressed only in primate. So I wonder if you can comment on that. Yeah, because I don't think I see like primate for the MPS.
Fantastic question, Sidi. No, to my knowledge, there are no non-human primate models of Sanfilippo. So the testing that does occur in primates, and it has occurred in primates, is more about safety, rather than efficacy. But I think, it's a good point that you don't necessarily know that if you're testing in a completely different model system, like a fly, for instance, that it's necessarily going to be effective in a mammalian system, and especially in a human. So that's the importance probably of having a couple of steps where you do test these things, and I guess using knowledge of biochemistry in the different species and conservation of genes and pathways, so that you don't embark on something in a species that is never going to have relevance in a person. Yeah, a great question.
Can I jump in and ask my question? Because it's still on large animal models. I think I read a guidance from the FDA, the US Drug Authority, that they were no longer going to require large animal models for preclinical trial approvals. Is that realistic? Do we think that will really happen? I don't know what you just said about the sort of the large brain, being there's normal dogs and normal primates out there that you can test distribution of a drug, for example, in the brain. But is it realistic that the drug authorities are going to go straight from a mouse to a human trial, for example?
Yeah, I'm not sure whether Nick can help me out here, but the N-acetyl Leucine, has that been in a large animal model?
Thanks, and thanks, Kim, for the talk. And I was going to ask this same question as to what role the regulatory bodies serve in either supporting development of models or, as Lisa is suggesting, facilitating translation in the absence of large animal models. To my knowledge, the N-acetyl Leucine has not been in a large animal model, nor indeed have a number of the gene therapies that have progressed through to human clinical trial. And indeed, increasingly, with a focus on development of N=1 and rare, ultra-rare conditions, there is some pressure, or at least support, perhaps is a better characterization, to drive that translation direct to human trials in the absence of large animal models. But I think you've highlighted some very critical reasons why, or deficiencies in that process that we may need to consider from a scientific perspective, and indeed a drug development perspective through your talk today. But it's certainly a changing space, as Lisa recognises. Should we be driven by the regulatory bodies is another question altogether.
So I actually had almost exactly the same question as Siti, because my brain was going immediately to primates, because that's traditionally been so important, and I hadn't actually heard of that, dropping the large animal models, because I would have thought it still is a critical step, as you outlined, for the dogs. And then I was wondering, when you were talking about the dogs and that they're really, those models, those colonies are really under pressure, and they might disappear. I was wondering whether ISA has a role to play here, you were talking yesterday, Lisa, about the registry, but, with that alliance, is there a role to play on a, international scale to somehow, maintain those models, whether they're, frozen, and then can be used when needed or something.
I think that's a really good point, and something in terms of resources and being able to, pay for the support of a ongoing dog colony, for example. ISA doesn't have at this stage significant funding resources. We haven't pooled funds or gathered funds from elsewhere, but in terms of advocacy and trying to find funds, if that is considered a priority, if the research community and regulators, or primarily the research community, feel that they need those models and they're going to need them in the future, then that's something that ISA as a community, a global community, could look at advocating for and supporting in some way. That's a good point. We'll add it to our discussion list.
And I can just add that the National MPS Society in the US has done and continues to support in, very significant, but significant, not necessarily money terms, but significant in terms of maintaining dog colonies and some of the dog research. Matthew's a big advocate for them. I can definitely see their value. And so they've been integral in keeping some of those models going in the US.
This is a comment really about AI, and I know AI is starting to be used in perhaps diagnosis, actually teaching or trying to develop algorithms to use clinically for detection. And one of the issues that has arisen just in meetings that I've been to is that the continual change and the sophistication of the AI. And so you can put in questions and then you get different answers. I guess it's just like the chat GPT. You can, put in lots of questions. And I guess the way you're thinking about using AI is different to that. You're not actually going to ask questions, but rather use AI in designing. Would that?
I'm just putting AI out there as a solution. I'm not an AI expert, but people are certainly starting to use it to, well, one of the examples is to look at the pathogenicity of the different missense mutations in the NAGLU. So there, I hope you don't get a different answer every time you.
That's what was raised in this discussion that I was listening to. That there is that potential, and then depending what AI platform you're using, there are differences, and then with the different generations of AI. But it might be that in that circumstance, they're trying to train to answer a question for detection purposes. It's not in the LSD arena, but
maybe that's something I can throw to some of the people in the audience and who I know work with AI. So Carissa, I know, works with AI. I'm not sure if anybody from the Bardi Lab is happy to speak to that. I'm not sure if Carissa.
So it's very dependent on what the question is. And the thing with AI and machine learning is you need so much data as input to train these models to then get to the accuracy that you want. So that's why with diagnosis, like with the mammogram images and stuff, they're putting in thousands to hundreds of thousands of images for training to get that kind of 99% accuracy. And so for the context of Sanfilippo, I think in people, I don't think there's enough kids in the world to get this kind of sample size that you need to get a really accurate machine learning model. And then would you? Yeah, if diagnosis is the question, but then is for disease mechanisms. So like in the omics realm, that's where you can generate the large numbers that you need to be able to train an AI to classify a mutant versus wild type and then get a treatment, like similar to what the Bardi Lab presented yesterday. Like that's an excellent use of machine learning. But yeah, for people, I worry that there's not enough kids in the world to be able to use it in a more clinical context, at least for Sanfilippo, maybe in a broader LSD childhood dementia site. But yeah, the Sanfilippo, yeah.
And so like any other model, it has some advantages and disadvantages, and areas you want to use it and areas you can't use it.
Yeah.
Yeah.
Yeah,
Thanks,
to add
Tracy.
Angel. Angel's also very knowledgeable in this
Great.
field.
Serena's online and she can unmute herself. And
Oh, perfect. Thanks, Serena.
Can you guys hear me?
We can hear you.
yes.
Yeah. Awesome. Yeah, I think I had a similar thought to Marissa, where I don't know about, the benefit of AI and like the diagnosis and things like that. But honestly, your talk has really highlighted how many models there are, and using AI to combine data and metrics across models might be an extremely valuable thing. So as there's so many different models and different metrics, and there's so much data being generated, I really could see the benefit in AI coming through and then starting to combine and finding commonalities and things like that, and really identifying what's out there.
Fantastic. Thank you for that, Serena. And Lisa?
I got all excited then because I had a thought. To follow on from Serena's point, the question I was going to ask next was about therapeutic windows. That's something that came up in the process of putting together the roadmap and identifying therapeutic windows, but more specifically than that, identifying therapeutic windows for specific types of therapeutics. There might be different windows for different targets, and thinking about all of the models that we've got and the things that have been tested in models. Maybe there's a way to start answering that question by analysing data across multiple different models and multiple different agents or pathways that we want to target with drugs. I don't know if that makes sense or would even be possible.
Yeah, good question. I'm not an AI. Oh, Chris is,
Yeah.
bleeping.
So I definitely think, so me and Angel have been talking a bit with the Australian Institute for Machine Learning. So this was all with her zebrafish behaviour, but I think we should go definitely have a chat with them because they are the experts in this field and they would understand what our kind of data sets look like and whether machine learning is applicable, because sometimes people just get excited by it's AI, it's machine, it's the hot thing, but actually traditional statistical methods are actually more appropriate for more certain questions. So maybe we should go see if we can set up a meeting. There's one guy I have in mind who's like he's a geneticist by trade, has moved into AI. So he's got like that mental health background.
Yeah. Well, maybe this is something that we could set up as some sort of, collaboration, a workshop involving families, patient societies, researchers, clinicians, everybody who wants to come listen and contribute. And I guess what we'd want to start with is, what are the questions we want answered, and can AI help us?
Yeah.
Or will it be a hindrance and send us off, down the garden path? In that case, we don't want it. But what are the questions we as a field want answered?
Totally agree.
Yeah. Yeah. Maybe if I can just take us on a slightly different path. I'm wondering for the family members, I didn't go into everything the models can do and everything we measure in the model systems, but if you were to want some questions answered about what the disease means for you and for your family, what are those, what would you like to see measured? And here, it obviously entirely up to you, but is there a symptom that you think is the most important thing we should be measuring?
With everything that you guys are studying, is there some way we've seen the behaviour, but is there some way that you could just branch off a little group to work on how to fix that behaviour? Sleep is a killer.
Yeah.
And I know my experience, behaviour doesn't last forever, but the sleep does. Lack of sleep does.
Yeah, there have been people who've attempted to look at sleep.
As Jordan's got older, no, I wouldn't say that. He becomes a little bit more tired and off balance if he hasn't had enough sleep. But no, the behaviour doesn't say pain changes his behaviour. But as younger, I don't think anything stopped it when he was younger.
So sleep is a big one you'd like to see measured in model systems that can measure it.
We've never had sleep. Never.
Okay, then that's important.
Yeah.
And pain?
Pain, we see his signals. So finding someone to listen besides, oh, it's end of life, it's just part of the condition, suck it up. Yeah, pain, you have to find the right doctor. But we can get pain meds are a pain in the butt because they want to use all the newest stuff. And we found that a lot of the older medication works better.
Okay.
So that sleep is huge.
Okay, good to know. Yeah.
I don't know if this is possible, but it's obvious, like when the children are treated or animal models are treated at an earlier age, that it has the best outcome. And when they're treated at an older age, they still clinically appear to be cured, but obviously they still suffer degeneration. So what is the mechanism that's still continuing to cause this effect?
Yeah, no, that's a very good question. Very good question. I think when Mark was talking earlier about, potentially modifying, say, inflammatory responses, that might be possible in advanced disease stages. But if neurons and their connections have been lost, that's going to be something that's very challenging to contemplate any sort of reversal of. Yeah. All right. Well, if we are all done with questions, maybe one outcome of the session is that maybe we think about arranging some sort of get together with anyone who's interested in contributing to the questions we want answered for the Sanfilippo field by artificial intelligence and machine learning processes. Then, maybe that's something, Lisa, that we can think about getting going in the future. Yeah.
Yes, definitely. I think getting a bit of a network together and tackling some of those big questions would be an exciting prospect.
And bringing in outside experts who don't know about Sanfilippo but know all about what different types of machine learning can do, and cross-pollinate.
Yeah, sounds like a good plan.
All right. Thank you.
Thank you very much indeed, Kim, for guiding that very interesting discussion, and everybody for contributing. We're almost at the end. I'm going to do a few thank yous and wrap up shortly. But before I do, I'd like to invite Teresa and Bala, if you're still feeling keen, to come and give us a few last thoughts and words on how you found the last couple of days and what it has meant to you.
Thanks, Lisa. Hi, everyone. My name's Teresa Lloyd. I'm a mum to two boys with Sanfilippo. I didn't think I was going to get emotional, but anyway, Ethan is 10 and Ronan is 8. So I just wanted to say thank you to everyone that's been involved with this year's symposium, and thank you to the Sanfilippo Children's Foundation for organising yet another fantastic event. You've all heard from other Sanfilippo parents today and yesterday, so Sarah and Gillian, and you'll hear from Bala, but basically about how small our world becomes after this devastating diagnosis. But what really is clearly evident is that our Sanfilippo world is not so small. This symposium has shown us that there's a lot of very smart people in this room who all seem to ask that they think that they ask silly questions or dumb questions, but you guys, it's amazing how smart you all are. And also people online that are with us and sharing this experience that are helping to expand our world through the research and studies in lots of multiple areas as well. So thank you to you all and to all your hard work that you're doing in solving Sanfilippo. But it does give me the confidence that sometime in the future, a parent or parents that are with a newly diagnosed child will not be told to go home and love them. But instead, here's a treatment, here's a cure. That's what gives me hope. Thank you.
Hello, everyone. I'm Bala. I'm Naumika's dad, a beautiful and happy 5-year-old girl who was here briefly yesterday before she turned all the tables over. So I firstly want to thank Sanfilippo Foundation, Karen and Lisa for creating a great platform of sharing ideas. At the moment, I'm having information overload, but it is when we come to sessions like these, all the sleepless nights or all the stress from dealing with behaviors and all the physical exhaustion, it goes back into the rearview mirror, and we are filled with hope, courage, and strength to keep going forward. Yesterday I was following all the presentations, all the wonderful PowerPoints, and okay, that seems doable. When can we have it? So yeah, I know. I mean, as I look around at the tables filled with families, researchers, and scientists, we are all connected via Sanfilippo, and yeah, we are all here to give every child who has Sanfilippo hope and life with comfort and joy that we can offer. Yeah, I mean, I'm filled with nothing but gratitude and appreciation for all the brilliant work. Whether it may be the liquid nanoparticle therapy or the delivery of enzyme therapy or the drug screening and the machine learning for predicting the neuroprotective thing, or for replacing the microglia. That really caught my attention. Replacing the diseased microglia with genetically modified ones. Wow, that was brilliant. Even the zebrafish models. Yeah, that was awesome. It almost makes me feel that we are so close to a world where, yes, we can treat and cure Sanfilippo. Bring it on. Yeah. So yeah. Having said all that, this is me being a desperate parent, eagerly waiting for a cure, and a Star Wars fan. Let's get to warp speed, and may the force be with you all. Thank you.
Oh, that's a hard act to follow. Thank you so much, Teresa and Bala, for those fantastic words. And I absolutely echo that. I work with all of you researchers every day. It's my job and my pleasure to work with researchers and assist collaborations. And it always, comes to a real head of excitement and hope and enjoyment in the work that we do, the serious work that we do to try and get to warp speed and solve Sanfilippo. Having these gatherings and hearing all of these ideas and the enthusiasm and excitement from you as researchers really is exciting. So it's been a fantastic 2 days. Thank you to absolutely everybody who has been involved, from everybody online and everybody in the room and everybody who helped to bring this symposium together. It's been really fantastic and absolute pleasure to work with you all. And just some specific thank yous that I must do. Thank you again to SAMRI and to the SAMRI team for hosting us here. It's big shame that Cedric was unwell and not able to be here and be part of it, but we appreciate him facilitating the use of this space and his wonderful team who helped us out. And also, again, thank you to the CommBank Staff Fund and Rohan for facilitating that grant that actually made it possible for the families to be here, and some of the speakers as well. A thank you again to the planning committee who helped us shape this program and bring all of these people together, to City and to Kim, Cedric, Michael Chan and Jan Kaslin, who couldn't be here but were very helpful in bringing together the program, and Teresa, who contributed in the planning as well. Special thanks. Sorry, I've lost my lost my train of thought. Thank you especially to everybody who helped in the room. we have a little thank you gift for the team who have been in the back corner there. They absolutely nailed their presentation up here at the front, but they've also nailed the tech work at the back of the room as well. So Paris, Ella, Alejandra, Zarina, thank you very much again for your help. And we do have a little gift of thank you for you to share nicely, and make sure that you share with the whole lab as well. Thank you very much again for making everything run so smoothly at the back of the room. I'm going to do a thank you in advance to the Lardelli lab, Carissa in particular, who's rallied the teams and told them to clean up the lab. Because the lab at the University of Adelaide, they're going to give the families and us, I'm really excited about it as well, see their lab, a tour of their lab after lunch, and we're really excited about that. And we get to see the fish, and Jamie, who's a zebrafish enthusiast from back last year, is particularly excited. So we also have a thank you in advance gift, again, share nicely. And we're looking forward to that tour. So thank you very much. I know a lot goes into, it's particularly tidying up the lab, but planning what you're going to talk about and show, it's a lot of work as well. Thank you again to Emma and to your mum for Emma for facilitating the connection, but to your mum for the wonderful, beautiful quilts that she brought along, for being a huge part of yesterday. It was so fantastic talking to her, and I know we've all enjoyed meeting Heather and enjoying her quilts and the families who get to take a quilt home. I also wanted to do a little shout out to acknowledge Louise O'Keefe, who passed away earlier this year. You heard about the fruit flies, and a lot of the work in Sanfilippo has been done by Louise and her team, and developing fruit fly models. She's contributed a huge amount to the field. And I think that today is an appropriate forum to acknowledge her and the work that she did, and to pass on our condolences to her family and friends and everybody that's known and worked with her. I know lots of people in this room know and work with her as well. So thank you, Louise. Who else do I need to thank? A special thank you to Kim. Big nod at the back there. Kim, you've played a huge part today. You've chaired multiple sessions, facilitated many talks. You are a huge force in the field, and much of what we do would not be possible without the work that you have done, in the lead up and in terms of your lab work, but also supporting the coming together in this forum. So thank you very much, Kim. Nick, thank you for showing up, and as well when you, we know how busy you are, and, and you are in pain at the moment, I know, so thank you very much for being here and for your contributions to the field and to the discussions and to supporting the families and answering all of their questions, and generally, just being a huge part of the field and the work that we do. I think that's all the thank yous. Have I thanked everyone that I need to thank? Oh, yes. Well, thank you to all of the Sanfilippo Children's Foundation team, because quietly in the background, Rebecca, who's in the cheery yellow today, and Karen, who you've all met, and Danny Sinney, who you all know from all the emails she sends and all of the things she does. She is the biggest quiet achiever that I've ever met, and gets everything done so efficiently. And this wouldn't have happened without Danny. She's done a huge amount of logistics and quietly getting everything ready behind the scenes. So a big thank you. And I think that's it. Just thank you for everybody for being here. I covered everything. Please stay in touch. Continue sending your questions and discussion. If you think of something after you've left that contributes to any of the discussions that have taken place today, you want to be connected with anyone that you've heard speak, please keep in touch with us and we can see what we can do. We want to keep the conversations going. I think I've covered everything. Thank you so much, Kim. Yes. For those online who didn't know, that was a thank you for me. Thank you very much, Kim. Thank you. That's it. Thank you to everyone online. Thank you for being there. Thank you, everyone in the room. We will see you all next year, hopefully. Thank you very much.